So next on our virtual stage, I'm very happy to announce a person that has founded 4 spin-offs. 3 of them are highly successful, and she is a professor of Bionanoscience at the University of Oxford. So thank you for being here and having the next keynote, Molly Stevens.
Thank you so much. I hope you can hear me all right, and see my slides. Yeah, I'm delighted to speak to you today, and thank you for inviting me. I'm an academic, actually, as my primary job, but very involved in translation, and particularly involved in translation in the field of developing materials for different types of biomedical applications. So this is my research group. You can see in the top right, it's a really, really interdisciplinary group. We've got surgeons, chemists, physicists, engineers, and people from all around the world. And it's a pretty big group because we do a lot of different projects, and also we really need all those different disciplines to come together for the kind of scientific challenges that we're addressing. Now, quite unusually, our lab does do some very wide spanning from fundamental science through to application, but very often we find actually that the fundamental insights we get from fundamental science help us to think about new inventions and that we then take forward for translation. And we founded a number of different spin-out companies. I'll mention some of them today. Some are still at very early stages, and others are more developed, and in fact also have approval for various products. So we moved in April to the Kavli Institute at the University of Oxford. Before that, I was 20 years as a professor at Imperial College. And the reason we moved to the Kavli is this is an absolutely state-of-the-art environment for this kind of interdisciplinary research that we want to do. And we're right at the interface with medicine. And so that interface between engineering and medicine is really, really key to address key clinical problems with state-of-the-art technology. And you can see here a sort of schematic of some of the different areas we research in. And so we make materials that interact with biology in interesting ways. And that might be, for example, for tissue repair or advanced therapeutics, or could be for early disease detection, or indeed instrumentation that can be used for new types of characterization. So today, I'll be talking about some of our spin-out companies, but before I do that, I want to just also make the point that you can also structure the academic research centers in a way that are going to really help with translation as well. And so what I want to show on this slide is an example of a hub we set up in the UK, which is called the UK Regenerative Medicine Programme, Smart Materials Hub. And this was to develop materials for regenerative medicine. It was a well-funded program that ran for 10 years. That was the cycle of the program. I was serving as the director. And we brought together 11 different universities across the UK to make lots of different biomaterials that you could use, for example, to repair the eye, the liver, the musculoskeletal system, and so on. And what was unusual about this hub is that, unlike a normal academic center, right from the beginning, I set it up with panels that could help us with safety and immunology questions, that could help us with questions around manufacturing and scale-up, and essentially a lot of advice on regulatory pathways too. And that's been very, very successful in terms of leading to a lot of academic science, but also many spin-out companies, as well as several clinical trials now being planned. So the other thing with this hub is right from day one, the researchers that come in are trained to think about target product profiles. So what is going to be the specs that are needed for a material to really make it in the clinic? And that is just something that is not done so frequently within academic settings. So I'm going to show a little bit about some of the things we've been working on and translating. First point I want to make is our group works on lots of different types of nanoparticles for advanced therapeutics. So these might be made from lipids, for example, or polymers, as well as more biological nanoparticles. And one of the things we were struggling with, and the whole field struggles with this, was the lack of tools available to really characterize at the single particle level what was going on with our nanoparticle formulations. And so a few years ago, I set the challenge to my group, let's develop a technique that can really give us insight into single particle chemistry. And so we developed a technique that involved an optical trapping, and we could then measure Raman spectroscopy, which is a technique that will tell us about chemical fingerprinting, and we could do that for individual nanoparticles in an automated way. And this was very, very powerful, because we could now start to look at formulations, and we could trap these particles, understand their chemistry, understand where the drug was loaded, for example, understand what size they were, look also at real-time dynamic reaction monitoring. And this was all enabled by coding and software that we've written in-house. And so we published this in this academic paper, but before doing that, we patented this technology. And at the time that we published this paper, this is what we would call a breadboard setup. So it was essentially within a darkroom, so only the physicists within my room, within my group could use it, and it was really needed, specialist training. And so what we've done since that time is we secured some money, initially just some pump priming money, to convert this technology, this academic technology, into an instrument that even someone non-trained could use at the touch of a button. So really to see how we could get this technology adopted by a much wider community. This led to this founding of this company called Sparta Biodiscovery. So it stands for Single Particle Automated Raman Trapping Analysis. We can analyze complex formulations of particles, trap them individually, and with Raman spectroscopy, reveal their chemical fingerprint. And you can do that and work out the heterogeneity within your formulation, and that can also help you with better rational design. And you can look at very complex mixtures as well and deconvolute all the information about your particles in there right down at the single particle level in an automated way. And this has been a very exciting journey for us. This instrument has now received CE mark approval across Europe. It's used by many tens of pharma companies as well as academic groups. And you can see here some of the types of particles that we've looked at, and also some of the things we can do. We can really understand how drugs are loading, whether proteins are forming on the surface. That's going to tell us about how these things are going to perform potentially in the body, how they're going to degrade. Lots of really, really interesting questions for pharmaceutical industry, but also for looking at biological particles like exosomes and telling, for example, if you have an early cancer. And so this investment to this company started with some angel investors that we're very grateful for, who believed in us right from the beginning. And once we had de-risked the technology slightly, we were able to receive a large strategic investment from a company called Sartorius, and have been working very closely with them to roll out this technology. You can see the latest version of the machine there now on that table on the left. That is CE mark approved across Europe. And these are some of our great team off at various conferences and, various academic institutions as well, where these machines are being demoed. In fact, our early access program has been so successful that all the machines from that have now been allocated to customers, and we are now moving on to the next phase of our production. Coming back to this again, I want to say just a little bit about the formulations themselves. And so another very exciting translational journey that we've been on has been being part of this wonderful company that was founded just a few years ago, called Nanovation Therapeutics, working on lipid nanoparticles. And this is for delivery of mRNA and really kind of cutting edge within the field of genetic medicines. And Peter Cullis is the chair of this company. You can see the leadership team on this slide. And this involves 6 different academics actually within the company. The lead base is in Canada. And this has been developing incredible IP that's now within the company around new types of lipid nanoparticles that have improved circulation properties and potentially then really, really exciting applications of these in a number of different diseases. And just about 3 weeks ago, we announced that Nanovation Therapeutics was signing a deal with Novo Nordisk for $600 million to look at a range of different clinical targets. So this is a really exciting time for this company, and it's been wonderful to be part of this collaborative team. I'll give just a slide about a much newer company now. This is a company I'm also very excited about. It was founded between myself and another academic that was with me at Imperial, Stefano Angeletti Uberti, and he is a computational scientist. And between us, we're using this computational drug discovery approach and AI with some of the experimental approaches from my group to essentially design new types of binders that can be used to treat different diseases. And this took a slightly different route to the capitalization that we took with the previous two companies. And this went through an accelerator program in the States called Y Combinator. And at the end of that, Y Combinator, quite a short sort of few months incubation period, raised its first round from various VC that you can see on the slide there. And it will be, it's well funded now for a little while. And at some point later next year, we'll raise some more funds. But in the meantime, what Nanograb has done, and this is the scientists in Nanograb have essentially all come from Stefano and my academic groups, is Nanograb's been able to show that they can design binders that really have fantastic selectivity. And this is because that we can use a multivalent approach, and we can decrease non-specific interactions. And we can even target different ligands at once to get synergistic binding on, for example, real biological cells. So this is going to be very interesting, I think, for new types of drug delivery that are more selective, and also potentially in some nanoparticle targeting approaches as well. So then I'll move on to talk a little bit about disease detection, which is another big area my group works on. And we have a few different companies we founded in this area. This company, Signature Biosciences, was funded I'm mostly just talking about companies funded in the last 3 years or so here. But Signature was a company that we founded to make precision diagnostics more accessible, because we had actually discovered some fantastic technology within the academic lab. So John Gertz, who was my postdoc at the time, and myself were co-inventors on a technology called the PCR architecture, which is a new kind of biosensing approach with new reporters and also some machine learning components. And then Celestin was on an entrepreneurship program at Imperial, worked with us for a year, and then became our CEO. And then we've expanded the team to various employees that you can see on the right, as well as some really fantastic advisors. And so this is our team. This also went through Y Combinator and then raised a round after that. And then we got a really generous, fantastic grant from a UK funding agency called Innovate UK, together with my lab at the University of Oxford. And this has enabled us to push forward the science in a very collaborative way between the university and the company. And we have exchange also of personnel between those two companies, as approved, of course, by the university. And Signature will soon be closing its next funding round. So there are already plans to finish that by the end of 2024 and then raise our Series A in 2025. So what Signature does is make much better breast cancer prognosis tests. We know that there is a real problem in that many women don't benefit from chemo post-cancer surgery, and you need a prognosis test to guide treatment decision. At the moment, those tests are performed in a centralized lab. They're really expensive. They take 3 weeks, $4,000, and you're flying samples across the world. With our technology, we can decentralize the testing, and we can make this done within local labs. We can measure 21 genes at once due to our algorithm. Our first product is going to be called OncoSignature Breast. And this is a really, really fantastic technology that is faster, cheaper, can be integrated, and we will be providing better value propositions for US and EU, as well as opening up new markets for Asia and rest of the world. You can see our first target is breast cancer, but we have various other clinical partnerships also in the pipeline. As well as the technology performing really well now, it's been demonstrated for breast cancer, but also some other disease applications. We have a wonderful quote here from the Deputy President of the Uni Cancer Network, essentially validating the promise of this completely new type of test. In terms of thinking about the wider community, because I know many people will be thinking of spinning out companies and how do you sort of set yourself up for the most possible support in those areas. I applied for this chair in engineering technologies 4 years ago now from the Royal Academy of Engineering. I was delighted to be selected as one of 6 candidates for this. And this is really a chair that covers some of my salary, so I can focus a bit more time thinking on translational research. And as part of that, I was able to select a mentor who was a fellow of the Royal Academy of Engineering. And I selected Hermann Hauser, that many of you will know, who's, of course, from Austria and a fantastic person, as well as fantastically successful in the translational space. And Herman's given me a huge amount of advice. And so these mentorships, I think, are really important if you're thinking of embarking on this journey. I've also had lots of support from my former postdoc advisor, Bob Langer, who's always, as well as Herman, at the end of a phone call if I need any advice on anything to do with thinking about translation and companies. And so setting yourself up for having as much support as possible, because this is a difficult journey and these things can really help. And so through that chair in engineering, I've been really thinking about the future of healthcare and the fact that it's going to need to be multi-omic. It's going to need to integrate AI. And through that, I've also been successful in securing funding from the Bioinnovation Institute in Copenhagen, which is part of the Novo Nordisk, funded by the Novo Nordisk Foundation. And they help you to incubate a scientific concept, de-risk it through their generous funding, and then at the end of the 3-year investment, form a company. And we are going to be spinning out this company called Dandelion Diagnostics, which is a diagnostic platform that is applied to longitudinal sensing of many diseases. And we've shown really great results in a first trial here for multiple sclerosis. And this is based on a lot of research from my group. You can see some of the journals at the bottom there, where we combine different types of sensors with AI algorithms and spectroscopy. Then the final slide I wanted to show, which is just kind of thinking about where do we go from here? So the companies I've talked about have either launched or are about to launch. And this company, this is not a company, sorry, this center, Q Biomed that I'm showing on this slide, is a new initiative that we just had funded. It's going to start in December. It's going to be a 5-year program, very generously funded from the UK. This is a £24 million national quantum research hub. It is the first hub in the UK, and indeed really going to be sort of at the forefront worldwide, where we are applying quantum sensing to biomedical related problems. And this is very exciting. There's great capabilities in terms of new types of early disease diagnosis and treatment approaches. It is absolutely the interface of cutting edge science and medical establishments. So we're involving many medical hospitals in this, as well as 17 industrial partners, government regulators, etc. So this is one of the next initiatives we're working on. I'll be the deputy director for this new exciting hub. And then I'll just finish there and give thanks to the group, the funders, And it's been a very, very exciting journey so far, and I'm happy to share any learnings from that. Thank you.
Much for these insights. And to the audience, there is still the possibility to ask questions in the chat. The first question is if it's allowed to share the slides. Would it be possible to share the slides afterwards? Maybe I directly ask that.
So I think they're going to be posted online as a recording. So yeah.
This is also a great hint for the moment. We are recording as well, which means you can have a look at the videos as well. Afterwards, they will be published on the website. If you have further questions, feel free to ask. I would start right through with the first one. Thank you, Molly Stevens, for the insights. And could you maybe just to sum up, what would you say are the three main learnings you got from your spin-off career?
Oh gosh, I mean, I think so. I think there's several things. So one, you know, as in an academic group as well, team is very, very important. So getting the right team. However, it's not just about team. So you also need to have the right market. And certainly I can give an example with Signature, for example. When we first launched the company, the beachhead market that we were going for was not as appealing to investors. And it took us a little while actually to think about and discover the application where our technology would be absolutely perfect, which turns out to be this OncoSignature breast application where we have this 21 gene signature. And our technology is just fantastic for that, but also there's a massive medical need for that. There's a great opportunity to displace a product that is way more expensive and less functional. And so this, you know, getting the market right is really important. Having good IP, of course, if you're spinning out from the university, making sure your IP is really solid and you have good IP lawyers, and then building your network. And that's something that, you know, Herman and Bob and others have helped me with a lot. So building that network of people that you know within the entrepreneurial space, as well as investors. I didn't mention it in my talk, but I've also just taken on a role at Oxford University as a university-wide champion for women in diversity and entrepreneurship as well. And so having that kind of community of people is really important.
Yeah, thank you very much. And as today's conference is about the topic about the navigation through the IP terms with universities, so there's also the question, chat, if you could describe your experiences with the TTOs. How was it?
Yeah, so I think what would benefit all universities really is to make that process as fast as possible, actually, because time is a really important currency as well. I mean, my previous companies were founded through Imperial College, and the next two that I'm doing are going to be through Oxford. One of the things that I've seen at Oxford that I'm really excited about is they have what's called an express license deal, which has essentially a 6-week time frame on it. So you can move really quickly, and that's very, very important. So I would say to people, when you're thinking about IP terms with universities, you know, the terms are one thing. Obviously, you've got to make sure the royalties are sensible and not going to cripple the company and all these kind of things like that. But speed is really important as well. So don't go just for the kind of bottom line on the money side, but make sure there's a commitment to working within a time frame that is going to be acceptable to your company.
Mm-hmm. Thank you for these insights. Very interesting about also about the express part. There are 2 more questions currently in the chat, and the next one would be about the public funding support. Which role did it play?
Huge. So, you know, we fundamentally develop most of our technologies from the academic lab, right? And something like Sparta, for example, was several years of R&D development before we even published that first Nature Comms paper and before we published it. And, you know, that never would have been possible without the grants. I think getting the funding that then helps you go a little bit more translational is something that governments could invest more in, because there's been studies, for example, performed and written up, I think, in places like the FT that have shown that for every $1 that you invest in this kind of bridge funding bit, you know, when you're getting from kind of fundamental to higher level concepts, that $1 will bring you back $29 of value. So to me, it's an absolute no-brainer that we should be investing in that kind of sweet spot of making technology go from academic concepts into higher TRL levels. With Sparta, I can tell you that we published our Nature Comms paper, and it was that breadboard setup. And we then received, thankfully, a very small translational grant. It was only about, I can't remember the exact amount, but on the region of about 100K or so. And that was enough to turn that technology from the breadboard setup into what looked like an instrument, right? That instrument box. And that 100K investment was then what gave people the kind of prototype that meant that we then got investors. And I think if we had not been able to get that little bit of bridge funding, then the whole rest of that story would have been a lot more difficult to take forward. So, you know, public funding is hugely important. And I think pots of money that can help in accelerating that kind of bridge area, something I would really encourage as well.
Mm-hmm. And you mentioned, we talked about the public funding, and you mentioned the investors. There's also a question about the investors, especially in your field. What key factors do investors typically consider essential for exit strategies for the diagnostic companies?
I mean, I think diagnostics is slightly different to therapeutics, and obviously I've been involved in companies in both of those areas. Diagnostics typically, I think you really need to get that right market application, you know, and that's why I think a company like Signature has a great chance of success, because it's identified, you know, the perfect application. And the margins, for example, on many diagnostics are less than on therapeutics, but also the route to approval is a little easier and less costly as well. So I think I don't know exactly what investors are looking for, but thankfully we have received investments. So it's probably a combination of team, application, and technology.
Thank you very much for sharing your spin-off experience and your insights. Thank you for that, Molly Stevens.
Thank you so much. Thanks.